So we know that the neurons that make up the central nervous system, that being the brain and the spinal cord, are really important. But what we haven't looked at is how these are protected. So in this video, we'll be looking at protection of the central nervous system. We need to protect our central nervous system because of its absolutely critical nature in terms of our survival, but it's also needing to be protected because it's delicate and soft tissue. So the neurons that make up the brain and the spinal cord are delicate and soft, and even the slightest of pressures can actually injure the neurons. So as a result, there's a number of ways that we can try and protect them from damage. Firstly, the cranium or the skull obviously offer protective outer coating. We have underneath the cranium and between the brain a number of layers of membranes called meninges. And then within those layers of membranes, or within the meninges, we have circulating cerebrospinal fluid, which is critically important for protecting the brain, as we'll talk about later. And then we have things such as a blood-brain barrier, which prevents nasties from getting into the brain, as well as the circle of Willis, which tries to work to overcome blockages in blood supply. What we're going to do in this video is discuss each of these in just a little bit more detail. Let's start with the meninges. Okay, I seem to have lost my heading. So this is the meninges here. And they're just protective layers that sit between the skull. So this is the skull here with the hypodermis and the epidermis layer above it. And our meninges are membranes that sit between the skull and then the underlying nervous tissue. So this is the brain here. Okay, well, what are these meninges? There's actually three of them. There's something called a dura mater, arachnoid mater, and a pia mater. So you can think of this as a tough membrane. This is a tough one. This one here has spider-like bits, so this is a spider one. And then the pia mater is a really soft inner layer. Let's talk about what these do. So if I can draw your attention over to this side, our dura mater, as I said, sits just under the bony tissue of our skull. Just kind of outlined it in green there. And really, although it's made up of two individual layers, that's not important. What is important is the fact that it's just really tough tissue, so it's strong. It's tough and it provides protection. Directly underneath that dura mater, we have a space. And that space is known as the subdural, just underneath the dura, the subdural space. And that's important in its own right because the subdural space, which I'm just trying to outline in red here, has a number of blood vessels within it. And those blood vessels are important for reabsorbing fluid within the brain. We'll talk about that in the next slide. Sitting underneath the subdural space is something called the arachnoid mater. The arachnoid mater. And that's the white one that I'm now kind of going over with black. And we've got some villi on the arachnoid mater. But a number of projections which look a bit like a little bit like a spider web. And consequently it's called the arachnoid mater. Sitting underneath the arachnoid mater is the area where the fluid of the brain that protects the brain, the cerebrospinal fluid, actually circulates under this in this subarachnoid space before we get to the pia mater, which is the glad wrap, really thin, soft membrane that just kind of covers the brain, as I've just shown there. Okay, I got a bit tongue-tied, so what I'm trying to say is we've got our dura mater, our tough layer on the outside. Directly under that is our subdural space, which contains some blood vessels that we'll talk about in a minute. We've got our arachnoid mater. Underneath that is a subarachnoid space, which is filled with cerebrospinal fluid, and then a soft layer that clings to the brain like glad wrap, being the pia mater at the bottom. So I've mentioned the word cerebrospinal fluid. What is that? Well, cerebrospinal fluid is a clear fluid, and it basically just circulates both within and around the brain, and also within and around the spinal cord. This fluid is made from plasma. It's a capillary filtrate derived from plasma, so it takes out the bits that are necessary from the plasma and sends back the bits that it doesn't need. But the important thing is that the cerebrospinal fluid helps protect the brain in a number of ways. It offers buoyancy, which means basically it reduces the weight of the brain by 97%. If you can imagine that the brain is a bowl of jelly, if you turn that bowl upside down and pop the jelly on the plate, the first thing you notice is that the top of the jelly squashes the bottom of the jelly, and suddenly you end up with a bigger, flatter pile of jelly than what you had in the bowl. Similar for the brain. The top of the brain would squash the bottom of the brain if it wasn't for buoyancy offered by the cerebrospinal fluid. So this basically prevents the top of the brain crushing the inferior brain tissue. But the cerebrospinal fluid also offers shock absorption properties. So if, for instance, you had a blow to the back of the head, your brain would move forward in your skull, and if it wasn't for the cerebrospinal fluid, it would knock straight into the actual skull at the front, or the frontal bone. Luckily for us, we have cerebrospinal fluid, which prevents this in most cases. The cerebrospinal fluid, its last job is it helps nourish the brain and also remove some waste. What I want to do in the next slide is talk about where this fluid comes from and where it circulates. This is a little summary for you if you want to come back and study that, but let's look at it with a visual aid. So our brain actually consists of our nervous tissue, our neurons and our glia, but it does have a number of spaces within it, and these spaces are called ventricles. So we've got a ventricle here, and there must be other ventricles around. Fourth ventricle, there's 4 ventricles, number of ventricles, which are basically just open spaces. And these ventricles are lined by something called a choroid plexus. I'm just kind of trying to draw over the top of that at the moment. So these choroid plexuses, they've got, we could go on for hours about what they are, but what they do, and what's important to you, is that they produce cerebrospinal fluid. So the choroid plexus produces the cerebrospinal fluid, it enters the ventricles, and then flows within the brain, down the central canal of the spinal cord, all the way around the brain areas, and then sits within the subarachnoid space that I'm just tracing out now until it comes to a arachnoid villi. At the level of the villi, that filtrate can actually come in. The cerebrospinal fluid now enters the subdural space. In the subdural space, you'll remember there are a number of vessels which allow reabsorption of that fluid so that more fluid can then be replaced. Why would we need more fluid to continue to flow around? Well, that's how we deliver our nutrients and get rid of our wastes. So, start in our ventricles, cerebrospinal fluid travels around in the subarachnoid space, goes through a villi, enters the subdural space, and eventually is reabsorbed. So that's cerebrospinal fluid. I did mention the blood-brain barrier was important for keeping out toxins. It certainly does this, so it provides a protective mechanism by regulating what gets to the brain. And as a result of that, it provides a stable environment for the brain. The blood-brain barrier just basically means that the outside lining of the capillaries becomes harder to penetrate. So this is a capillary anywhere else in the body. Basically, we have the lumen of the capillary and then the endothelial cells that make up the capillary on the outside. But you can see it's a pretty flimsy looking structure, and because of the fact that we've got endothelial cells joining together, sometimes we can get toxins and other things that we don't want getting out. They just get out, they sneak out between the gaps between the endothelial cells. In the brain, we've still got our capillary with our endothelial cells, but we've got a bit of added reinforcement in the shape of astrocytes, which, as you may remember, are a type of glial cell. So here's our astrocytes. Basically what they do is they reinforce the capillary structure, and they help these endothelial cells to form tight junctions so that nothing can get out, so that less can get out. So the blood-brain barrier is formed by glial cells, or astrocytes, encouraging the endothelial cells to form tight junctions, preventing the unwanted toxins or perhaps unwanted molecules from getting in. So it decreases permeability. And as a result, the blood-brain barrier becomes selectively permeable, keeps out metabolic wastes, proteins, certain toxins, and most drugs. Well, that's all very well, but how do we get the blood to the brain in the first place? We don't just want to keep out the bad things, but we want to be able to let in the good things. And so therefore we need a constant supply of blood to the brain. I don't want you to remember the details here. It's not important to know what the name of the arteries are, but rather that we have arteries that come up the back of the brain, as well as an artery that comes up kind of more towards the front and supplies the middle of the brain as well. The question is, what would happen if we blocked one of these arteries here? it would mean there would be no blood supply here and no blood supply there. So we want to be able to try and avoid this situation, and that's why we call in the help of the circle of Willis, otherwise known as the cerebral arterial circle. And this is all about providing a constant blood supply to the brain. So this is a picture looking at the bottom of the brain. What you can see here and what I'm trying to chase out is in fact the circle of Willis. Basically, if we have a blood, I can't show you because of the 3D nature of this, but if we had blood supply coming to the front of the brain and blood supply coming to the back of the brain, if one of these was blocked or occluded, it would mean that the rest of the tissue would suffer. But because of the circle of Willis, when one of these is blocked, if that's blocked there, we still continue to get blood from here, and it circulates around, allowing it to supply all the other regions of the blood of the brain from there. So take-home message, basically the circle of Willis provides alternate routes for blood to reach the cerebrum if a vessel becomes blocked, thereby providing constant blood supply.